Welcome to BIOL102 Introductory Plant Biology students. As you know, I am talking about the internal structure of plants, and today we are going to finish with this internal structure of plants. Today I am going to talk about the secondary growth. This is PowerPoint number 4 in our series on structure of plants. So let us start with the internal structure of plants, that is the that is secondary growth. Now if you have seen this slide, if you look at this slide, you must have seen a log, and I hope that some of you will recall that what you see here, my pointer is here, what you see here are the growth rings. Today we are going to talk about these growth rings, also called annual rings, and they are formed in the tree of a plant, in the trees of a plant. And in the biology lab 331, which is now called Dr. Rehan Siddiqui Plant Anatomy Lab, there are these logs, transfer section of the wood of a tree, which are lying next to the main door. So when you come back to the campus, you must have a look if you have not looked at it before, and you can see these growth rings or annual rings. Okay, let us go to this first slide, and it says what are two types of growth. Earlier you have learned, I have told you that there are two types of growth: primary growth and the secondary growth. And these two types of growth, these two types of meristems are responsible for these two types of growth. We call them primary meristem and the secondary meristem. Primary meristem and the secondary meristem. Primary meristem are the apical meristems, which are located at the tip of root and at the tip of shoot. You are familiar with these apical meristems. You have already studied them. They cause primary, growth, and they're found in all plants. And the results in primary growth, increase in length takes place because of this primary growth. But in addition to this primary growth, in some plants, there is also secondary growth. And this secondary growth is responsible because of the presence of secondary meristems. As you will learn, there are two types of secondary meristem. One of them is called vascular cambium, and the other one is called cork cambium. And these secondary meristems are present in gymnosperms. These are the cone-bearing plants. Gymnosperm, you will study them later on, but you are familiar with cheed ka taraf, pinus is a gymnosperm. They are found in the northern parts of Pakistan, and those of you who have been to Murree, Kawan, Sawat, you have seen these plants, trees with needle-shaped leaves, the gymnosperm. So this is where it is found. And woody dicots, not all dicots, but woody dicots. There are some dicots you have studied which are herbaceous dicots, but there are others which are trees which you have seen in the botanical gardens. Most of the trees that you saw in the botanical gardens are woody dicots. And this secondary growth results in increase in girth, increase in diameter. They increase in diameter. Increase in diameter takes place because of this secondary growth. Okay, so today we are going to learn how this secondary growth takes place and what is the tissue that is formed because of secondary growth. There are two types of tissues which are formed by the secondary growth, and this is secondary xylem and this secondary phloem. And in addition, at the periphery of the tree, there is also formation of a tissue which is called periderm. You are familiar with this word bark. So bark or periderm is formed on the outside of a tree, and you have seen a bark when you were looking at these different trees in the botanical garden. Okay, now this is a good slide, and here if I can make it big, I don't know how I can make it big, but you can see this apical meristem. These are the primary meristems, and there are three subapical meristems: protoderm, procambium, and the ground meristem. Now, from the leftover procambium, you have the formation of the vascular cambium, and this vascular cambium will form secondary xylem. Another name for secondary xylem is wood, and it will also form secondary phloem. It will form secondary xylem and secondary phloem. And the case of cork cambium, it will form the cork. It will form the cork, and it will form secondary cortex. So this is how these meristems are: primary meristems and the secondary meristem. You have studied them earlier. So here this tell you about the primary plant body, and you can see that primary plant body is as a result of this primary meristem. This blue thing that you are seeing is the shoot apical meristem. It is present at the tip. It is also present in the axil of the leaf in buds. This is where it is present. So this causes increase in length, and this is the only meristem which is found in the case of herbaceous plants where secondary growth does not take place. But in gymnosperms and woody dicots, in addition to this primary meristem, there are also lateral meristems or secondary meristems. So this red thing that you see are the secondary meristem which are present in the stem and is present in the leaf. So this is what this slide shows. Apical meristem make primary shoot increase in length, and lateral meristem makes secondary increase in girth or diameter. So here he is talking again about secondary growth, and he tells you that this results in increase in girth or diameter, and woody plants have secondary growth, and the herbaceous plants have little or no secondary growth. It is present, secondary growth is present in woody plants. Here again, he is talking about the two main types of secondary meristem, the vascular cambium and the cork cambium. Vascular cambium forms secondary xylem and secondary phloem, and cork cambium makes the periderm on the outside. And today we are going to study how this happens. So here, to tell you how secondary growth takes place, first of all, you have to bring to your mind the structure of a dicot stem. And here I told you that in between, say, the primary xylem and primary phloem, there is this patch of vascular cambium. This vascular cambium, which is present in between the primary xylem and primary phloem, this is called fascicular, Fasciular, fascicular cambium. This is called fascicular cambium. And the first step that you have in the case of secondary growth is, by the way, this secondary growth takes place in the growing season. And in our country, the growing season starts in spring, and then it continues in summer. You know, in monsoon, there is a lot of rain. So growing period is spring and summer, when there is a lot of rainfall and there are moderate temperature. So it becomes active in spring. The vascular cambium. And then what happens is that in the beginning, it is in between the vascular bundles, but then this vascular cambium, now you can see this yellow ring. This yellow ring is the vascular cambium. It is also formed in between the vascular bundle, and now you have a complete ring of vascular cambium. Now this one, which is originally present between the primary xylem and primary phloem, this is called fascicular cambium. And the one which is formed later on in between the two vascular bundles, this is called interfascicular cambium. So you have the formation of a complete ring of a vascular cambium. What happens next is that it will become active, and these are black and white diagram, but still you can see in this diagram there is only fascicular cambium. Here you have a ring of vascular cambium. Here you have a ring of vascular cambium. What will happen to this vascular cambium? It will now start dividing. And this diagram shows how the vascular cambium cells divide. Here is the color scheme. This purple thing that you can see are the vascular cambium cells. So they will divide toward the outside, and they will divide toward the inside. This type of meristem, which divides in two directions, outside and inside, this is called bifacial. By outside, I mean toward the epidermis side. By inside, I mean toward the center of the stem or pith side. So it will form cells toward the outside and it will form cells toward the inside. The cells which are formed toward the outside, they will differentiate into secondary phloem. They will differentiate into secondary phloem. And the cells which are formed toward the inside, they will differentiate into secondary xylem. So by the activity of vascular cambium, secondary xylem and secondary phloem are formed. Secondary xylem toward the inside and secondary phloem toward the outside. This is what happened. Now it so happens that secondary xylem that is formed is retained year after year after year. What happens is it starts forming in spring and then it continues in summer and then in autumn it stops. It stops. In winter there is no growth. Next year it will again become active and you will have this secondary xylem formation. Secondary xylem is retained, but secondary phloem, the old secondary phloem is periodically removed from the outside of a tree. So here you have vascular cambium. This green thing is vascular cambium. This is secondary phloem that is formed. And here this blue thing is the secondary xylem that is formed year after year after year. And here you have to concentrate here and look at these cells of the secondary xylem. And you will find that early in the season, you cannot see this arrow, but it says early xylem. It says early xylem. So this is xylem that is formed. Yes, I want to introduce one term. I think I have already told you, but you have to remember this, that another name for secondary xylem is wood. Another name for secondary xylem is wood. Wood is secondary xylem. So this is the wood or secondary xylem that is formed early in the season. This is called spring wood. This is also called early wood or early xylem. Late in the season, you have this formation of this wood, and this wood is different from spring wood. These cells are, thinner wall. They have thinner wall, but the cells which are formed in the summer, they are thicker walls. They are the thick walled cells. So thin walled cells, thick walled cell. Again, next year there will be thin walled cell, then thick walled cell. So one spring wood and summer wood, or one early wood and late wood, this will form one annual ring. This is how annual rings are formed. This is what he's saying, how tree rings are formed because of these differences between the early wood and the late wood. So this shows how this secondary growth takes place. The secondary xylem is retained, and year after year you have the formation of accumulation of this secondary xylem toward the inside, and you have formation of these rings. So xylem remain in place, this is what he is saying, and continues to function for many years. And this results in increase in the diameter of the stem. And now after many years, you find that in a tree you can see two types of wood. There is a wood which is toward the outside. This is called sapwood. And there is a wood toward the inside, and this is called hardwood. So sapwood, I will show you in the lab. If you ever come to the lab, inshallah, then I will show you this difference between sapwood and hardwood, but you can see in these slides also. Sapwood is the outer wood. It is pale yellow in color, and this is functional. The hardwood is the inner wood, and it is darker in color, and it becomes non-functional. It becomes non-functional, non-conducting because of deposit of number of these substances. So sapwood and hardwood are the two terms you have to learn. Okay, so here in this slide, you can see these annual rings. Now, when you will look at this wood, you have to first locate where is vascular cambium. So my pointer here is at the vascular cambium. And from here to here is 1 year growth. From here to here is the 2nd year growth. From here to here is the 3rd year growth. So you can say that this is a 3-year-old stem. He has magnified this. So here is the vascular cambium. Here is the vascular cambium. And from vascular cambium, from here to here is 1 year growth. Here to here is the 2nd year growth. From here to here is the 3rd year growth. So this is 3 years growth. Now, if you go toward the outside, then you will find that only the functional phloem, the recently formed phloem, will remain here, and this is secondary phloem. And secondary phloem, along with the tissue that you are going to study next, will form what is called bark of a tree. So you have seen the bark of a tree. This is present on the outside of a tree, and we will talk about this when we will finish with this vascular cambium activity. Here again, you can see this is the outer wood, which I said is pale yellow in color. It is functional. This is the inner wood, which is darker in color, and it becomes non-functional. So this is secondary xylem. This is secondary xylem. And of course, you can see these growth rings in the sapwood and the heartwood. And here, this red thing that you see is the vascular cambium. This is the vascular cambium. And this vascular cambium will form secondary xylem toward the inside, and this is the secondary phloem that is formed toward the outside. And outside the secondary phloem, there is formation of periderm. And this periderm, along with this secondary phloem, is called bark. Okay. Here you can see another section of the wood, and you can see that it has two annual rings. This is one and this is the second. So it has two annual rings. This is vascular cambium. On the outside of the vascular cambium is secondary phloem, and outside secondary phloem is the bark of a tree. Here, this difference between sapwood and heartwood is very clear. You can see this wood toward the outside. This is the sapwood. This is paler in color. This is on the outside, and this is functional. And toward the inside is the heartwood, H-E-A-R-T, heartwood. It is darker in color. It is toward the inside, and it becomes non-functional. And then this is the vascular cambium, and outside vascular cambium, this region is the bark. Most of the tree is made up of secondary xylem or wood. 90% or more is made up of secondary xylem. Here again, you can see a section of the woody stem. This is first year growth, this is second year growth, this is the third year growth, and this is that vascular cambium. Outside this is the secondary phloem, and then the rest is the periderm. So this is another section of the woody stem. Okay. Now, after this, we come to this secondary type of meristem, which is called core cambium. You know, outside the secondary phloem are parenchyma cells of the cortex, and in these parenchyma cells of the cortex, there is formation of a secondary meristem, which is called core cambium. And this core cambium will result in the formation of periderm. And there are three components that are formed. Core cambium also divide like the vascular cambium, and it forms cells toward the outside, and it forms cells toward the inside. The cells that are formed toward the outside, I think this diagram will tell you about the activity of this secondary, this core cambium. So core cambium is also called periderm. It will form cells toward the outside, and the cells which are formed toward the outside will develop into cork, and the cells which are formed toward the inside will develop into secondary cortex. So this is the core cambium. This is the core cambium, and it has formed cells toward the outside, and it has formed cells toward the inside. The cells which are formed toward the outside will develop into core, and the cells which are formed toward the inside will develop into secondary cortex. So now I have these bullet point slides, and you can, I think, understand this because I have already gone through this. Okay, this is the bark of a tree. You have already seen different types of barks, but let us see what he says in these bullet points. I have already told you this, so this will be repetition. Gymnosperms and woody dicots have secondary meristem. Secondary meristem is called, one of the secondary meristem is called vascular cambium. This is the one which results in the formation of wood. Okay, so look at the cross section of dicot stem, and in between primary xylem and primary phloem, there is a patch of vascular cambium. This is called fascicular cambium, and then there is formation of interfascicular. The word is interfascicular, not intervascular. Interfascicular, INTERFASCULAR. interfascicular cambium. Okay. Fascicular cambium is vascular cambium is bifacial, which means that it will form cells toward the outside and cells toward the inside. The cells which are formed toward the outside will differentiate into secondary phloem, and the cells which are formed toward the inside will differentiate into secondary xylem. Secondary xylem is retained, but older secondary phloem is not retained. Only the recently formed secondary phloem is retained. Okay, the appearance of these rings that you have in the secondary xylem, these are called annual rings or growth rings, and you can find out the age of a tree by counting these annual rings. And this branch of plant botany, plant biology and botany is called dendrochronology. I have told you this. With the help of these rings, you can find out the age of a tree, and you can also find out about about the climate in the past. So it has a lot of application, dendrochronology. Okay, another name for secondary xylem is wood, and there are two types of wood, as I have told you, sapwood and the heartwood. Look at the slide of a log, and you can see again sapwood and hardwood, and then the wood that is formed early in the season is called spring wood, and the wood or early wood, and the wood that is formed later on is called summer wood or late wood. And you can see, I have told you, I have shown you this section in which you can see the difference between early wood and the late wood. Because of this difference in early wood and late wood, there is appearance of annual rings or growth rings. Now we come to the outside of the tree. There is this periderm, and in this periderm is again this periderm or core cambium. Core cambium is produced, and this core cambium will form cells toward the outside. The cells which are formed toward the outside will develop into cork or phellum, and the cells that are formed toward the inside will develop into secondary cortex or phloderm. And here you can see the bark. You have seen different types of bark yourself in the botanical garden. This is formed because of the activity of cork cambium, which is also secondary meristem. So I have already shown you this slide. So in the bottom slide, you can see the core cambium, and core cambium is forming cork toward the outside, as you can see in this slide, and it is forming secondary cortex or phloderm toward the inside. Okay. In this periderm, there are a number of these loose areas or holes, just like stomata, and these are called lenticels. So you have to find out the definition of lenticels from the book also. So these are loose areas in the periderm, and they are for the exchange of gases. Function of lenticels is exchange of gases. And this is the end of this slides. This is the end of this slide. I hope that you have understood these. I hope that you have understood these various the secondary growth. I hope that you have understood the secondary growth, and you have got an idea of how this secondary growth takes place. You have got an idea of how this secondary growth takes place. So this is the end of these slides.